Today, at least one person in this room cannot enjoy a piece of cake with gluten. And approximately 5 people in this room cannot enjoy a nice Gruyere cheese sandwich and a nice cappuccino as breakfast. For those who are addressed, probably asking why it is me, why it is have to be me. And you know what, I might have an answer for that. But before, I would like to invite you for a couple of million years of journey to search for the answers back in our ancestors. So here in this world map, you see the potential migration paths of our human ancestors. Based on the fossil records, we know that we migrated out of Africa about 50,000 years ago. And we immensely reshaped the biosphere. And there were mainly two major drivers for that. And then those two things are our genetic code and our diet. So our genetic code is probably one of the best places to look for those differences or uniqueness about humans. Just want to give you one example. We share 99% similarity with our closest living relatives, chimpanzees. And then that leaves us 1% that is uniquely human DNA. So this is probably where we should look at when we are searching for human uniqueness. Secondly, our diet is pretty much unique. We learn how to control the fire. We learn how to cook. Not any other animals did. And then by this way, we integrated like pre-digested, sterile, and highly nutritious food in our diet. And more recently, as human beings, we learn how to domesticate the animals and how to use their milk and meat products. So as you could see, our gut has these amazing capabilities to adjust these major significant life changes. And then in my research question, I'm basically asking the same, what is unique about our human gut? And then to find out what is unique about our human guts that adapted these major life changes, we need to compare human and our closest living relatives, chimpanzees. So to do this experiment, it's not that easy. So I might need to get multiple biopsies, like from anyone in this room. Or I might need to do this with the chimpanzees. In either case, it's pretty complicated. Probably nobody here want to be part of such an experiment. Multiple surgeries, 10 years long. No. And then the chimpanzees are endangered species. It's also very impossible to offer such experiments. But I have a solution that can make both you and chimpanzees happy. So how about if I say I can make mini organs in the lab? So, and to make this mini organs, what I need is a tiny skin biopsy from anyone in this room again. And I can take the skin cells back to my lab. I can reprogram them to their stem cell state. And here in the stem cells you see on these Petri dishes have the potential to make all the cell types in your body. And if I give them the right conditions, right nutrients, they can make one of my favorite organs. I think by now you know this, which is small intestine and our guts. And I can make hundreds of them. And these mini organs basically represents your genetic and dietary background. So just the first question is, how small are these mini guts? So here you see one example of 4-month-old human mini gut that is about 2 millimeters in size, which is about a grain of rice. And then I see some faces right now. If this tiny thing could do the same job what my gut is doing, like can it really digest the food? Can it protect me from bacteria? Let's have a look. So to demonstrate to you how this mini organ grows, I'm going to show you the very first 5 days of this mini gut. For simplicity, we label these cells with green, with red. Every red dot is one single cell, and then the green is the outer layer of the cells. So then you see they start to divide, they start to self-organize, and about day 2, 2.5, they start to make this lumen-like structure. So you can imagine this as like in 3D, like a sphere or ball. So this is basically representing your gut tube. So here, basically, when we give them some nutrients, so they can basically digest them and absorb them. And they are so clever, they can generate mucus, so they can entrap the bacteria or viruses when we injected them. And finally, they can sense the sugar levels, like by generating some hormones. So they also understand how much food is too much. So the next question is, you are asking right now, can you apply the same recipes that you make your mini guts to chimpanzees? The answer is yes. So it took me a couple of years to find, but finally we generated the first chimpanzee intestinal organoids. That is again 1 millimeter in size. Here you see 1 month old mini gut that is derived a chimpanzee named Sandra. So now we are at the forefront of this research area where we can generate hundreds of mini guts from humans, chimpanzees, and other species. And by comparing them, we basically would like to find out what is unique about our human guts, so how they function. And then secondly, looking for a needle in a haystack in the treasure box of evolution, I would like to search for the answers for today's gut diseases. Like, can we find those answers? What is the origin of today's gut diseases, like lactose intolerance or colorectal cancer? Can we find those differences back in our ancestor and bring it to the future? So probably in a couple of years, you will see on the news there are gonna be some highlights saying that scientists generated mini guts from healthy and diseased individuals, and then now they can find a personalized cure and personalized therapies for our gut diseases. So by this way, hopefully, we do not have to replay evolution back and forth a couple of times, but we can focus our future challenges by curing the gut diseases. Thank you very much for your attention.